And with that, I think we are ready to rock. So I want to thank you all again for being here. Thank you for your support in this weird time. We're so excited to see all of your faces. And to kick us off, I want to introduce a very special guest, our own Allison Rempel-Brown. Allison.
Thank you, Aaliyah. Thank you all for being here. Excited to see your faces, as Amy pointed out earlier. And we hope to be together with you someday in the future. And so good morning to everybody. Allison Rempel-Brown, President and CEO of the Science Museum of Minnesota. Your support helps us to continue our mission, which is to turn on the science, inspire learning, inform policy, improve lives. And it helps us do science, which we do, so thank you. Now, my mother-in-law, Barbara Brown, was a science lover too. And in fact, she was a scientist who worked in the research division of the Field Museum for 50 years. And she was motivated when she was a younger woman by Rachel Carson's seminal environmental research in the book, Silent Spring. And Barbara and my father-in-law, Roger, helped us create this critical science position at the Science Museum of Minnesota last year. This position centers on birds, and she was an avid birder and unfortunately did not survive to meet our first chair. And this chair is going to help us continue our ongoing research and conservation efforts. So I am proud, so proud, to introduce our first Barbara Brown Chair of Ornithology, Dr. Catherine Earley. Dr. Earley directs the museum's biology department, which contains thousands of vertebrates, invertebrates, plants, and a few birds. In addition to her research and fieldwork, she will build on the Science Museum's existing avian collection, and she may share some of her ideas on this with us today. Dr. Earley comes to us from a postdoctoral position at the Florida Museum of Natural History and the University of Florida. She completed her PhD work in ecology and evolutionary biology at Ohio University. So join me in welcoming Dr. Earley. All right, we're waiting for her to come. We could all make bird sounds.
Well, thank you for that introduction. I am going to do a little introduction of myself, but take it further back to let you get to know who I am. I had an interest in animals from a very early age, and that was fostered by a supportive family, including grandparents, as my grandmother helping me hold the fish. That continued on into volunteering at the North Carolina Museum of Natural Sciences when I was in high school. And I continued to work there while I pursued my undergraduate degree in biology and Spanish at North Carolina State University. And so both of those are in Raleigh, North Carolina, where I grew up. As Allison said, I earned a PhD in ecology and evolutionary biology at Ohio University. And I just finished doing a postdoc on digitizing museum specimens, which we'll talk a lot about today, at the Florida Museum of Natural History and the University of Florida. And throughout my career, I've built experience doing research and presenting this at conferences, working in collections, and communicating science to non-academic audiences. And so as you can imagine, it's been quite a transition to move from Florida, where I was swimming in these beautiful blue springs, although just a tip, if you go to Florida, you only swim in springs like this or the ocean, otherwise you're running an alligator risk. To Minnesota, where we do not have alligator risks, but we do have snow in October. So I'm glad I saved my Ohio winter gear from grad school. I can tell it's not going to be enough to get through Minnesota, but I think it's time to do some shopping for some base layers. And in case you can't tell by the crinkled corners of my eyes in the snow picture because I'm wearing a mask, I am excited to be here in the snow and excited to be here in general because I'm thrilled to be working at the Science Museum of Minnesota and to be talking with y'all today. So I will, with that introduction, get into talking about my research. So I study the evolution of the brains of birds, which underpin all the behaviors that we see in birds like cattle egrets shown here. And I'm going to zoom in and show you the skull of this egret and kind of pop out the model of the brains that I use to study the evolution of their brains and behavior. And I do all of this research with digitized museum specimens from collections like the Science Museum of Minnesota's Biology Department. Now, other researchers have established that the size of a brain region relative to the size of the brain overall correlates with the importance of the function or behavior it mediates. And so this is a bird brain, and I've just pointed out some of the different functions that different parts of the brain are involved in. And because of this relationship, we should be able to glean information about the behaviors of extinct birds from their brains. And obviously with extinct birds, this is kind of the only way to get at their behavior since we can't observe them directly. And it's important to understand the brains and behavior of extinct birds because the diversity of birds we see alive today, like this loon, didn't come out of nowhere. First, you had the evolution of dinosaurs, specifically non-avian theropod dinosaurs, and then 150 million years of evolution between those dinosaurs and the birds we see today. And so if we only study modern day birds, we're missing a lot of the story of how we got this diversity. So how can we study the brains of extinct birds, since their brains unfortunately don't preserve in the fossil record? One option is to use CT scanning, which is like a 3D X-ray. So I take a museum specimen, put it in the scanner, and the scanner generates cross-sectional images of the hard tissues of the specimens. And I use these images to make a 3D model of the brain. You can also use this to make 3D models of any of the hard tissue anatomy or the spaces that are encased by the hard tissues. And the model of the brain generated from a CT scan is called a brain endocast. So that's these blue brain-shaped models you've been seeing throughout my presentation so far. And it represents the space in which the brain sat during life. But it's not the same as the brain. So this is an important question. Can you make inferences about a brain from something that really just represents it? Well, some of my work that was recently published and work by my colleagues has shown that in birds, the brain endocast is the same size and shape as the brain. And so we can actually be confident in our inferences about brain anatomy and behavior just from the brain endocast that we can get in fossils and modern day birds from their CT scans. This is how I was able to study the brain and behavior of a species of moa, which is a giant extinct flightless bird from New Zealand. So you can see me standing next to the skeleton of one at the Carnegie Museum of Natural History and a paleoart reconstruction of what they would have looked like when they lived. My work on this species showed that a vision-related region of the brain of this bird, which you can see here in a kind of half cut open moa skull, is extremely reduced, which suggests that they may have had decreased visual capabilities. And that research is highlighted in SMM's Science Superheroes exhibit. So if you haven't had a chance to visit that yet and you want to learn more, I recommend going to see that exhibit. Now, the research that I've been talking about and my other research is inextricably tied to the digitization of museum specimens. On the one hand, CT scanning inherently digitizes specimens, like this parrot that you can see on the left from the cover of an article that I co-authored. And on the other hand, the only way I'm able to identify specimens that I want to study at museums around the world is because they've digitized their specimen records and made them searchable in online databases like this one on the right from the Smithsonian. When you talk about the digitization of museum specimens, you can think in terms of digitizing the specimen itself, which is what I've done when I've CT scanned specimens, and also part of what we're doing by photographing Minnesota birds and mammals for our collaboration with the Bell Museum Natural History for their biodiversity atlas. And digitizing the specimen through photography, as you can see here, or 3D scans like the CT scans I use, also digitizes data embodied by the specimen, such as anatomy, color, and even the number of parasites the specimen had when it died. But digitization of specimens also involves digitizing the records and the data documented on specimen tags, which you can see here from that same specimen of northern flicker from the previous slide. And this is another important part of the work we're doing with the Bell Museum for their biodiversity atlas. This side of digitization forms the backbone for any digitization of the specimens themselves. It helps us to direct growth of the collection, like Allison talked about when she introduced me, and it digitizes tons of other useful data that researchers can use. So for example, the only way that researchers like me know to visit a museum's collection, so the only way researchers will know to come to the Science Museum of Minnesota or to request a loan of our specimens, is if the specimen records have been digitized into a searchable database. The simple question, what specimens do we have in our collections, can be the most important one to answer if you're interested in growing the research impact of your collection by having scientists publish on your specimens. And the specimen data also record where species occur, how this changes through time, and how this changes in response to human activities. So data like those, so the data that I showed on the tag of the northern flicker, allow us to make maps like this one, where the warm colors represent high biodiversity, so high numbers of species of animals and plants, and the cool colors represent low areas of biodiversity. Understanding how species have responded to environmental changes in the past is key to mitigating their responses to the changes that we're currently causing now in the future, and that are resulting in unprecedented rates of loss of biodiversity. But if you also map out where the world's natural history institutions occur, you'll notice a huge disparity between the number of species and the number of museums. Now, there's mounting evidence that people who are most qualified to lead the attempts to slow the impacts of human-caused changes to the environment are those who live in close relationship with the natural environment, often indigenous people. And yet these are the same people have often been excluded from museum and academic spaces. So digitizing the data from these specimen records can get critical information about biodiversity, where it occurs and how it changes, into the hands of the right people. So as a quick summary, digitization inspires creativity because it does result in these beautiful images of museum specimens and also enables cutting-edge research like my own. It allows us to teach classic scientific concepts, such as the shared developmental origins of the vertebrate forelimb, in novel ways that help build an understanding of the importance of museums early in the educational careers of learners. And so for this example, this is a three-dimensional collection of forelimbs, so arms, all digitized from museum specimens, where the same bones are colored the same, and help students understand the concept of homology. And digitization throws open the vaults of natural history housed in so many museums while also preserving the specimens for future generations. So with that, I am happy to take any questions either about my research on bird brain endocasts or on the digitization efforts I'll be leading in the biology department. And I'm going to stop sharing my screen so that I can see you all and see Aaliyah while she's asking questions.
Hi, everyone. Well, thank you so much, Dr. Earley. If you want to, everyone, you can go to your reactions bar and give her a hand. That was great. Oh, fantastic. Everyone's applauding. I'm so glad to see that. So Dr. Earley, thanks again and welcome so much to the museum. Clearly, you have a lot of experience and a lot of deep understanding of bird brains, and we were making little jokes about how bird brains, in fact, aren't that small, despite the popular recognition of it. Can you just maybe start? I'm going to remind everyone that if you have a question for Dr. Earley, you can go ahead and put that right into the chat, and you can either chat to everyone or you can just chat to me. I'm host one. So if you want to, either way, I will get your question. So I'm going to just send you all a note just reminding, chat to me here. But to kick off the conversation, it's such a specific topic with bird brains. How did you get interested in ornithology initially. You showed us some really fun slides of you as a kid, and I'd love to know a little bit more about how you found your way to this area of study.
Well, it was a little bit winding. So like I said, I always loved animals and museums. But I thought because I loved animals, I would have to become a vet to work with animals. So I didn't really see myself like working in a museum, even though I loved volunteering at the North Carolina Museum of Natural Sciences. So when I went to undergrad at NC State, part of the reason I picked it is because they have a vet school associated with it. And I took some pre-vet courses my first year and decided that that was not for me. Hadn't really made a decision on what else I'd want to do, but just kept taking courses. And my second year, I got into an Introduction to Dinosaurs course. And I just really loved it. Like, I think the ability to think about evolutionary questions and try to figure out, how we got the diversity of animals we see today, and especially the relationships between forms, like the anatomy and the function that different parts of animals carry out, and how that influences how different animals interact with their environment, was all really fascinating to me. So I asked the professor if he was teaching any more courses, and he was, so I kept taking the courses he taught. And eventually he asked me if I wanted to do some paid research in his lab as an undergraduate. And so I said yes, and I started doing that. And he actually was already looking at bird brains and bird brain endocasts. And so that's where I got my fascination for this topic. And I realized that there were still so many questions unanswered, especially that the first question that I talked about that I recently published on, if there is even a strong relationship between the brain and the endocast in birds, because that's something, it was an assumption that was underpinning all of our inferences about behavior in extinct birds. So yeah, that's how I guess a few taking the right courses and getting lucky, and also getting really lucky meeting my mentor through those courses. So he and I still talk and work on research papers, and now that he has kids, I read books to his kids over Zoom. So I think good mentorship is really the key to how I got to where I am.
Well, that is a really interesting path. Thanks for sharing that with us. And for all of you, I see lots of good chat questions coming in. So I'm going to start with the first one, which is from Barb Proschel, asking how much of SMM's collection has been digitized.
So in terms of having that backbone that I talked about, so getting the records digitized, not much. Almost. I think before the Biodiversity Atlas project started, which that predates me, I wasn't involved in writing the grant, but it's really great to come into a project that's already started and already kind of has a framework. So I think before that started, the answer would be none for the biology collection. I think anthropology is much further ahead in terms of having the collection digitized. So we have a lot of work to do, but it's also kind of exciting to be on the ground floor or something like that. I can do it the way I think will best serve the museum, rather than dealing with maybe some legacy databases that don't work the best. And I come from, so when I was doing my postdoc, I was at an institution that also houses something called iDigBio, which is a national resource for digitizing museum specimens and getting those records online. So I feel like I've had a lot of good training to be able to tackle this and kind of direct it. So the Minnesota Biodiversity Atlas project is just digitizing Minnesota birds and mammals, but that's a large chunk of our collection. I think most of our birds and mammals are from Minnesota. So once we get that done, we'll have, reptiles and amphibians, fishes, invertebrates, and plants left to do. Now, plants and invertebrates and fishes, actually, all of those groups are pretty diverse, so we probably have a lot of specimens in those groups too. But the Biodiversity Atlas is setting us up to have a large portion of it done.
Wonderful. Thank you for that. From David Lilja, how does the size of the Science Museum's bird collection compare to other museums?
Because we don't have the records digitized, I can't say in terms of numbers. I know the biology collection has about 170,000 specimens total. I think it's easier to talk about the strengths of the collection. So the Science Museum is really strong as a regional collection. We have a lot of specimens of the same species from different areas of Minnesota and the upper Midwest. And so that's really important for getting at questions of like regional variation and also variation through seasons. So I think that's a strength that museums like the Smithsonian, for example, which is somewhere I did a summer internship during my PhD, they don't have that strength. You know, they might have some areas that they are stronger in, but they're, you know, the National Museum of Natural History. So they kind of try to have more representation from all over the world. So I think museums like the Science Museum of Minnesota, our strengths are in the richness and depth of information that we have about the species that we do house rather than the number of species.
Thank you. So the next question I have is from Nita and Rick Lewis. It says, I read that with ice melt, they've discovered 800-plus-year-old penguins in Antarctica. Do you know anything more about the study?
I don't, but I love hearing that because, so when I talked about how brains don't preserve in the fossil record, unfortunately bird brains or bird bones, excuse me, also don't preserve super well because they are pretty thin and tend to get flattened. So like some of the most beautiful preservation that we have in the fossil record is flattened animals, including flattened birds, but you can't get the shape of the brain from those. So when I hear about something that's been preserved in ice that's now melted, that's great. Because yes, there is, you know, a bunch of pressure under ice too, but I think the chances of it being, of us being able to reconstruct the brain and 3D anatomy of specimens like that are much better. So I haven't heard about that specimen yet. I'm sure my undergrad mentor, who is along with doing bird brain things, is really a penguin specialist, has something to say about it, but I'm gonna look it up after this so I can find out who has the specimens and what they're planning to do with them.
So someone stumped Dr. Earley, but I'm sure we'll follow up in another presentation with her on that. From Rebecca Bond, is there a particular part of the brain that changed as birds evolved from two-legged ambulators to flying creatures?
Oh, this is a great question. I was just at a conference, a few weeks ago for the Society of Vertebrate Paleontology. So that's the scientific society that I've been involved with for the longest. And someone presented on new research where they take a bird, I think they were working with starlings because they're common and easily trainable. And they put a little helmet on there for PET scanning, and PET scanning, let's see, I don't want to get too much into it, but basically it lets you see which parts of the brain light up while birds are doing different things. And while, you know, anything that's wearing the PET scan helmet is doing different things. And so that's really cool work. It's really preliminary still, but that's showing which parts of the brain activate while birds are flying. Since that's so preliminary, I can talk about what I do know about the major changes that birds went through. So one thing is that most birds reduced their sense of smell from compared to non-avian theropod dinosaurs, so things like T-Rex. And you can see that reflected in the brain. The structure called the olfactory bulbs is pretty small in most birds, although there are exceptions like the kiwi, which uses scent along with touch at the tip of its bill to find food and basically can't see it all. And turkey vultures also have really good sense of smell. So when you see them flying over, they're actually smelling for their prey. And a region of the bird brain that expanded a lot is called the optic lobe, or the optic tectum if we're talking about the brain. And that's actually one of the regions I focused on when I was doing that ground truthing study. And so both of those tie to what we know about bird senses. We know that birds don't rely a lot on the sense of smell to navigate their environments, and they do rely a lot on vision, some more than others, but there's still a strong pressure to expand that part of the brain. So when you look at a bird brain compared to a non-avian theropod brain, the optic lobes are very expanded. And then also just in general, the cerebrum, so like the forebrain, which is the wrinkly part of our brains that, like, it takes up the most space in our skull also expanded in birds compared to non-avian theropods. And that's been hypothesized to be tied to kind of what you talked about the way you wrote your question about the change in how they navigate their environment. So flying instead of just being bipedal. But interestingly, some other research has shown that the non-avian theropods that birds were most closely related to were already on that trajectory of increasing brain size relative to body size before flight evolved. So it's kind of like they already had the brain they needed to fly, and then they started flying, which isn't what we would expect. So that's why it's always good to test our assumptions and do these kinds of studies.
All right, that was a great question and an interesting answer too. The next question comes from Sarah Lilja, and it's how will you and Dr. Kraka Gator be working together? It sounds like you have a lot of interest in common with Dr. Hastings.
Yeah, it's really great to have another paleontologist here because, I am an evolutionary biologist, but really they kind of bleed into paleontology because, like I said in one of my early slides, you can't study the evolution of modern day animals without understanding where they came from. And Alex also, he's more of a paleontologist, but also is an evolutionary biologist. So it's been great to see where there's overlap in our knowledge and also where we can each bring different things to the museum. Something we've been working on recently is with the STEM education group. We're both in the paleontology working group, and we've been serving as like expert advisors on the activities they've developed. So we helped them develop a Croctober activity, which obviously was more of Alex's thing, but I got to work on that as well. And we'll also be working with that group to adapt one of their lesson plans called Dinosaur Dentist that normally uses casts of the teeth of extinct animals and also modern day species that had the same diets to teach kids about like the specializations of different types of teeth for different types of food. And we'll be working with my former postdoc advisor at the Florida Museum of Natural History to replace those casts with virtual models from the CT scans of museum specimens that we were generating while I was a postdoc with him, and some CT scans of dinosaurs that I've been slowly tracking down because those aren't as easy to access because people don't like to put them online so much.
So the next question comes from Tom Molaski. Hello, Tom and Kathy. I love your garden backdrop. How can someone access the digital collections, not just at the Science Museum, but also at other museums?
That's a great question, and is really important. I talked about for my own research, as well as just I think for making museums feel more accessible to the public and to people who it might not be easy for them to go to museums. So one of them is iDigBio, so that national resource that I mentioned earlier for digitizing databases. They aggregate data from all databases all over the world that share their data with them. So if I want to search for common loon, and I want to know where I could find loon specimens, I can type in the species name to iDigBio, and it will show me the records for every specimen of loon that has been that the institution shared their information with iDigBio. So that's one of the goals for digitizing our database is we don't just want a database online on our website that people have to go to find our specimens. We also want it to be in iDigBio. So when researchers look for loon specimens, they know, oh, there's some right here in the Twin Cities. I don't need to go over to the Smithsonian to study loons. So that's one way for the records of the specimens and like the data around the specimens to be shared, and that's our plan going forward. The CT scans that I use for my own research are put on a website. We can put them on a few different websites, but the one I prefer is called Morphosource. And so when I publish on these, so like once I've done the science I wanted to do on these CT scans, I put them up on Morphosource and anyone can request to download them. And that helps us track how many people have downloaded scans of our specimens. They usually have to enter in information about what they want to use them for. And just from tracking the usage of the CT data from my postdoc, we know that about 50% of the people who request to download CT scans on MorphoSource are researchers, but then about 25% is for education and 25% is for art and just for other personal uses. So that's a way that people can actually directly engage with the data of the specimens themselves.
Great. Here's a question from Tim Eder. What drew you to the Science Museum of Minnesota?
Well, as you can probably tell, I love museums in general. And this job was open at the right time. Actually, technically a little bit early. I had 2 years on my postdoc and I only ended up doing 1. But when a job like this comes along, you don't drag your feet and say like, oh, well, maybe there will be another one in a year, because there won't. The museums are very, very special and people really like working at them. So people don't tend to leave museums very often. And when I was looking at researching at the institution when I was applying and doing my interview, I was blown away by the very real commitment to equity and justice issues that certainly, so when I applied, this would have been, I think maybe September was when the application was due. I think I interviewed in December. This was before George Floyd had been murdered and Ahmaud Arbery and Breonna Taylor. So before these equity issues were really at the forefront. Now, granted, there had been plenty of unprovoked murders of Black people and other people of color throughout America's history and even recently, but I think for whatever reason, those events really propelled these things to the forefront. And so now it feels like most institutions and organizations at least do lip service to these issues. But the fact that the Science Museum of Minnesota had a commitment that predated these things, and like I said, it seemed like a real commitment and not just lip service, that really drew me to it. And now that I've been here, I've been able to experience how true that commitment really is. And that's something that's important to me because as a woman in science, there weren't always a lot of us in that field. Like Barbara Brown was actually breaking some barriers by being involved at the Field Museum for as long as she was. And so I've seen, experience firsthand how important it is to have representation and also just to deal with the issues of like sexism and the issues that seem to come up when people of certain groups aren't represented in areas. And so I also obviously have a lot of privilege as a white person. And so it's been important to me to use that to break down some of the barriers to LGBTQ access to the field, because it can be very dangerous to do field work if you're a member of those groups, or to the access of people of color to our fields. Because while vertebrate paleontology, for example, we do have a better gender balance now, at least when you look at the level of students, it's still a pretty white field. And I know we're not getting all of the ideas, all of the good ideas in the world if we're still limiting access to certain groups of people. So it's a very long answer to say that I wanted to be in a museum, and this museum really impressed me with its commitment to issues that I feel strongly about.
Oh, that's a wonderful answer. Thank you for that. And also, I have to say, I think it goes really well back to your presentation about why digitization is so important by increasing access and access for people who can share new research and new science using the collections that you're accessing. So thank you for that. I have a question here from Allison Brown. What was the biggest surprise for you when you started working with our collection?
Ooh, biggest surprise. This is a tricky one. I think one of the things that has surprised me is how it's organized, which is kind of a boring answer, but like I really am interested in how things are organized, especially as we're looking at digitizing data and looking at use of the collection. So I'm not used to our collection. If you haven't been in, is our vault is all in one continuous room. And so you have anthropology in one section and then biology and paleontology, and they're clearly labeled and separate collections, but they're all in the same space. And so I'm used to like, when I was at the Smithsonian, obviously it's a huge building, and like if I wanted to go from the bird collection to another collection, there was a good chance I would get lost in the process because they're in very different parts of the building. So that's been interesting to be in a place where all the collections are together. Granted, there's, for certain types of specimens, like the wet specimens, alcohol preserved specimens are in their own room because alcohol is flammable, and you want that to be in a special vault where if something happens, it doesn't affect the rest of the specimens. And the bone specimens are also in a different room. I think because there are certain pests that are tied to them that you don't want getting into anthropology specimens, for example. But I think the fact that they're all in one room and there's so much connection really lends itself to connection and collaborations between the different disciplines that are represented by science at SMM. So it's something I'm still getting used to, but it's something that I like. I like that I don't have to go to a totally different part of the building to talk to the anthropologist or to talk to Dr. Kraka Gator about research, because all of our specimens are right next to each other and our offices are pretty close to each other too.
Well, that is a terrific answer. Thank you for that. Also, I think we're kind of wrapping up the end of our questions. Is there anybody who wanted to just unmute themselves and ask a question? If anyone's having issues with chat, now's your chance. I know there's some people who are calling in as well. Any others? Oh, it looks like the Bonds have a question, so we'll take that one.
Yes, thank you for this wonderful presentation. You had mentioned about the kiwis are unique as regards their olfactory skills, olfactory bulb. And are there other examples in Australia or New Zealand of birds that appear to be unique to that area, which might reflect, their geographic separation, as is the case with some of the other unique animals that we would be more familiar with from those regions?
That is a great question. And so I'm looking down when you guys are talking to me because the screen is a little bit offset from the camera. When I'm answering, I'll be looking up. So yes, there's a species of parrot called a Kakapo, endemic to New Zealand as well. And they don't have this olfactory specialization, but they are nocturnal, which is another trait of the kiwi. And they also are pretty flightless. They're pretty chunky parrot, and so they mostly use their beaks and feet to get around. And the fact that they are nocturnal means that they also have a reduction in their optic lobe, so that part of the brain that I talked about being reduced in kiwi and moa. And I'm not sure about Australia now that I think about it. I mean, there's certainly terrifying birds like cassowaries over there, but you don't necessarily see the same specialization. And if I had to guess, it would be that, so one of the reasons we think that New Zealand birds could evolve the way they did, which unfortunately also results in a lot of them being very vulnerable to invasive species, is that New Zealand, when these birds were evolving, didn't have any mammals on it, and there were a few bats, but no terrestrial mammals. So there weren't any mammalian predators of these birds. And so in that painting I showed of the moa, the big bird coming down was called a host eagle. So it was a giant eagle. And that was the only known predator of moa. They didn't have, tiny little rodents running around eating their eggs, for example. And in Australia, I do think they had the marsupial mammals. You would have giant megafauna, just like you would have like mammoths here. There were giant kangaroos and giant dingoes. So I think perhaps the birds couldn't be quite as helpless on Australia and make it through because they had those predators. But it's a really, really great question, because New Zealand does seem so special and unique in that way. And there's another related one would be Madagascar and the elephant bird. That's another giant extinct flightless bird, very much shaped like the moa. Has the moa beat just by a little bit for the largest egg that we know of. And they also seem to have a reduced optic lobe, not as much as the moa, but there does seem to be reduction. So this is actually a question that came up at the conference I was at a few weeks ago. So you are very, very cutting edge with your question, and something that I'm kind of hoping to pursue. It's got my wheels turning since that conference. This question of why you see reduction of the optic lobes in certain island birds, and looking at what other animals they evolved with, maybe dietary niches that they, so like what kind of diet they were eating too. So
Thank you.
yeah, thanks for the question. And I almost brought down our kiwi specimen, and I didn't. I'm kicking myself now because it's so it's so cute and beautiful. But if you're on if you follow us on social media, they posted it this week for Winged Wednesday. So there are some great photos of the kiwi mount that we have. And I think we're really lucky to have a kiwi specimen in our collection, because we, as the United States does, for a while we're getting as many kiwis as we wanted. They are endangered though, and they're actually also a sacred bird to many tribes of the Maori people. And so when conservation was really tanking of the kiwi, New Zealand and the Maori said, no, people can't just take our kiwi anymore. And now they have better, and I think more equitable rules in place. So now they do have systems where they will send a kiwi to a zoo, but if the kiwi that goes to a zoo in another country is first generation non-New Zealand, its remains have to go back to the Maori in New Zealand. So after a certain point, it might have been like the 70s, any kiwi specimens in collections are going to be like second or third generation or on specimens that were imported from New Zealand. So I think that was, when I learned that, was an interesting idea, because as museum curators, we're always wanting to make sure things go into collections, but it's also important to remember that there are other viewpoints about what should be done with both cultural objects and also natural history objects, and it's important to respect those and work with those as well.
Wonderful. Well, I want to thank everybody for all of your terrific questions, and I want to let you know you're going to have a lot of additional opportunities to see Dr. Earley in the coming weeks. In fact, we're going to follow up this conversation with an email giving you links to some of the other great bird resources that we already have on our website, and some upcoming exclusive content for our Facebook member page. So if you're not a member of that already, please do go ahead and find us and join. And with that said, I do want to thank you again for joining us. And I would like to just try something. If you can give me a quick show of hands using the reactions, who learned something today? Or a thumbs up. Oh, I love, look, I'm seeing all those terrific thumbs up. It's so great to see all of you again. Wonderful. And I also just want to acknowledge your support and thank you for everything that you've done for the Science Museum of Minnesota and for our programming in this challenging time. And remind you that next month, we will be doing another Coffee with a Curator event with Dr. Alex Hastings. And we're going to have him take you on a virtual dig. So he's going to show you how he scouts a dig site and figures out which is the right place to start a dig and to start some research. And he's always also got some cool artifacts to show you as well. So that's going to be on November 18th. We'll be sending you a note about that. And we're also going to send you, as I mentioned, a follow-up email that's going to have a lot of additional content about birds. And it also includes a survey. It really helps us if you can take a stab at filling that out, especially as we're transitioning to this virtual programming space. So thanks again to all of you. It's so wonderful to see each and every one of your faces. Thanks for your support of the Science Museum, and welcome Dr. Earley. Thanks, everyone. Hi.